My name is Sarah, and I'm honored to be part of the invited symposium of queer researchers at this year's conference. I'm definitely more comfortable giving presentations about my own research on baby bird growth and parental effects, but instead I'm sharing my perspective on how my queerness has shaped my research questions and how my research has informed my own identity over the last decade. I'm a queer, non-binary evolutionary ecologist. I recently defended my PhD at the University of Illinois at Urbana-Champaign. I've also spent the last 2 years as the chair of the Rainbow Lorikeets, a 2S LGBTQIA+ caucus of the American Ornithological Society and Out to Innovate. As part of my work on behalf of the lorikeets, I have publicly spoken about my queer identities and the way that they've influenced my well-being, my job prospects, and my sense of belonging in research spaces, especially as my life and the lives of other trans researchers have been impacted by recent legislation. A criticism I often receive in the face of this often terrifyingly personal work is that I should stick to the science and leave my identity out of my outreach and communication work. When I was invited to join the symposium, I decided to explore this idea a little bit more. Is it possible to just stick to the science? Biologists may often like to imagine ourselves as a kind of bias-free conduit of empirical reality. We collect data and use mathematical probabilities and models to rigorously assess alternative hypotheses. If done correctly, the hope is that we don't allow our own preferences, assumptions, and egos to bias our assessment of our data. Of course, proper experimental design and statistical analyses alone do not completely prevent the possibility that our collective scientific output is biased. The questions we choose to ask, the systems in which we ask those questions, and the emphases we place in our interpretation of our results are often not random. In practice, this means that our collective scientific output may be inadvertently skewed. Take, for example, recent work by Silas Fisher and colleagues assessing the bias in avian ecology studies in the USA and Canada. The researchers found that colorful, aesthetically pleasing birds were 3 times more likely to be studied than the least colorful birds. And birds were much more likely to be studied if their breeding ranges overlapped the location of universities. I personally have always studied birds within 10 minutes drive of my university office. I can collect more data with fewer research funds, and I can reduce my carbon footprint that way. But as Silas and their colleagues point out, has this implicit bias led to disparities in our knowledge of North American birds and their behavior? We scientists are not magical, bias-free machines. Our real lives and our perspectives shape the kind of research questions we pursue, the locations and systems where we ask those questions, and the way we present our results to the broader community. The best way to ensure that our implicit biases do not inadvertently contribute to knowledge gaps in our broader scientific understanding is to ask ourselves what unique perspectives we bring as researchers, question if it's influencing the way we perform our research, acknowledge the limitations of our own work and perspectives, and invite the participation and collaboration of researchers whose perspectives are different from our own. I don't want to speak for all of you, but acknowledging implicit bias was not a skill set taught to me in many biology classes. It's not something I find inherently easy to do. So for the rest of this presentation, I'm going to take a stab at it to ask how my identities may have shaped my work, and in turn how my work has influenced my identities. What was my perspective when I started performing independent research? Was this shaped by my identities? Well, I could count on one hand the number of openly queer students in my rural high school in Ohio. I grew up feeling like a weird outsider, told by members of my community that I was against nature, broken, sinful, and wrong. I found solace and supportive mentors in the Ohio Young Birders Club. Being part of a birding club did me absolutely no favors. In my high school social scene, but it did allow me to seek in birds the weirdos and the outlaws just like me. A bird outside of the range map printed in the field guide was a cause for celebration, an unexpected behavior at the feeder worthy of notes in my journal. I sought to learn about bird morphology and behavior, to memorize what the data show about how birds are, but also keep note of the individual birds that broke those rules, that went against their expected nature, the bird outlaws, just like me. As I trained to be an evolutionary ecologist in undergrad, I learned how to collect and interpret data, to be part of the scientific community that helped builds the data-informed backbone of expected animal behavior and physiology. But I also kept an eye out for the many individual animals that defied my averages and broke my expectations. For example, I was tasked with studying territory distributions in grasshopper sparrows spread unevenly across the prairies of Kansas. Testing my population genetics hypotheses led me to take blood samples from adults and nestling sparrows for genotyping. For the nestlings, I tried to take samples when they were 6 days old enough to have developed veins in their wings, but young enough I wasn't worried that I would force them out of the nest with my intrusion. In measuring those nestlings, I started to notice that they were not all the same size at day 6. Some sparrows were substantially larger than their siblings or conspecific sparrows at nearby nests. When I looked to the published literature on grasshopper sparrow growth, I got the average size of each bird at a given age, or maybe a range of sizes, but the birds I was observing were falling well outside of even those ranges. For my master's research then at Kansas State, I measured the growth of not only baby sparrows, but also dickcissels and eastern meadowlarks, finding that their growth rates varied substantially more than was previously reported in the literature. By the time they were old enough to leave the nest, some nestlings were 50% larger than conspecifics growing up mere meters away. Interspecific variation that could have some serious implications in a population that experiences extreme early life mortality bottlenecks. I connected this intraspecific growth variation to ecological drivers during my master's work at Kansas State University, and then moved to the University of Illinois to study the physiological mechanisms that give rise to this intraspecific variation in avian growth rates. So as a child, my queerness made me feel like I existed on the margins of my peer groups, breaking the expected rules on presentation and behavior. Did that draw me to the study of the individual birds that existed on the margins, who grew and developed in ways that didn't match those published expectations? If so, I'm grateful. Intraspecific variation is, after all, a key requirement for selection, and I'm immensely proud of my work connecting this variation to ecological drivers, physiological mechanisms, and evolutionary patterns. If my identity contributed to the field observations that I found fascinating and the questions that I chose to ask in my research, did it also work in the opposite direction? Did my research influence me? After all, I started doing academic field research in my first year of undergrad, which is notably an important time for many students to grow to understand themselves. I personally never felt like I needed biological observations to prove or justify my queerness, but given how often these so-called biological facts are used to justify discrimination against queer sexualities and gender identities, I did find it heartening to see publicized observations of organisms in same-sex pairs or individuals performing social roles that were not expected based on their chromosomal sex. I saw it in my own work too, a nest of grasshopper sparrows being raised by 2 adult females without an attending male partner, and female dickcissels displaying plumages typically associated with males. During my PhD, I fostered American robin eggs of known laying orders to test hypotheses about the effects of laying order on post-hatch growth. My efforts were frustrated by a pair of female robins who both laid eggs in a single nest. When I stopped by the nest to collect the day's freshly laid egg for this experiment, I consistently instead found 2 freshly laid eggs, preventing my use of the eggs for the fostering experiment. Queerness doesn't need to be biological for queer people to be protected, supported, and welcomed in our societies, but it was still meaningful for me as a queer person to see my own research subjects in same-sex pairs or challenging binary sex phenotype assumptions. Despite the emphasis of the symposium, queerness is not my only identity and not the only one that has interacted with my work. As I've navigated the last 20 years, I've collected a perplexing collection of health diagnoses, mental and physical illnesses that have been disabling in unexpected ways, like decreasing bone density in my early 20s that left me performing daily fieldwork tasks in walking casts, which I would not recommend. Like many scientists would, I tried to stay abreast of the scientific literature for each diagnosis, kept a list of all the genotypes associated with each diagnosis. Eventually that list was long enough, I sort of wondered if I just had the unluckiest genome possible. In my PhD then, I started studying maternal effects, the ways parental behaviors and phenotypes can influence offspring phenotypes. Much of my work focused on the hormones passed from the parent to the offspring through the egg yolk, a suite of 32 steroids. I started to map the hormone metabolic pathways and the physiological feedback loops that could link the presence of egg hormones to the observed nestling phenotypes. Much of the papers I read for this background information were mammalian or even human studies, and many dealt with the health problems that could occur when hormone pathways were atypical. In doing this research, again, research for my papers on bird yolk steroids, I came to suspect that many of my seemingly unrelated health issues were not unrelated genetic conditions, but perhaps the phenotypic expression of an endocrine problem. I brought this hypothesis to my doctors at the University Health Center, who agreed to run the test to check my hypothesis, and I learned that not only did I have a serious endocrine problem caused by a malfunctioning adrenal gland, but it was well on its way to causing irreversible and deadly damage to organ systems across my body. I didn't expect my dissertation work on bird egg hormones to directly contribute to saving my life, but my doctors assure me that it absolutely did, which is wild. Our identities have the potential to shape our research, and our research can shape our ever-changing identities in significant ways. This has the potential to negatively bias our collective research output if we leave these perspectives unacknowledged or if we construct research communities that don't represent diverse perspectives. At its most extreme, a biased scientific output can leave glaring holes in our understanding of the world. For example, the prevalence of same-sex pairings in some animal populations have been observed for centuries, but were downplayed to protect heteronormative narratives and ideals. Only in recent decades have researchers started to publicize the true extent of same-sex behavior in non-human animals and start to speculate about the evolution of a behavior that may be much more common than we previously thought. We need to understand our own perspectives, build diverse and inclusive research communities that can reach well beyond our personal biases, and continue to assess and reassess the ways our research questions and the conclusions we choose to emphasize are related to our identities. If not, we risk presenting understandings of the world that do not truly encompass empirical reality, and our scientific theories are worse off for it. In short, we can't just stick to the science, as if the science we perform is not influenced by who we are, our identities, our strengths, our weaknesses, our histories, and our perspectives. Scientists are people, and our scientific communities are strongest when we can include as many strengths and perspectives as possible to avoid biasing our results toward a single dominant narrative.